Okay, so 9.4 is about reproduction in plants, and more specifically, it's targeting flowering. So we're talking about flowering plants, which are angiosperms, and you need to connect this to what you know about angiosperms from Unit 5. As you can see, this part of the slide here, okay, there's a common view that flowering is influenced by biotic and abiotic factors. Now, connect the idea of what abiotic is in terms of what we know about ecology. So we're talking about non-living factor, and in particular, it's the day length, or as you come to notice later on in the slides, it's the night length which influences flowering. So abiotic factors which influence flowering is the amount of time a plant is spending in light. That's called photoperiodism, which we're going to unpack in quite a bit of detail. The other thing to mention is that what we've got in terms of the flowering plant is a mutualism that's going on. That is that two organisms, for example, the honeybee and the sunflower plant, have got a mutualistic relationship. You've got vectors of all sorts which are involved in moving pollen. Now remember, pollen is the male gamete, and what you want to do is encourage cross-pollination so that you've got sexual reproduction. So mutualism is an example of an abiotic factor, or any kind of vector that's moving pollen for you. So pollination is all about looking at essentially biotic factors. You've also got some organisms which are involved in the dispersal of the seed. So again, this would be a biotic factor. And if we look at these syllabus statements here, I want to emphasize in particular U1, first of all, up here. And you've got connections here which are transcription and translation, higher level stuff. So it's really important, especially when we come to talk about the flowering response, that you recognize that this Unit 9 is a higher level unit, and there's the expectation that you are linking and connecting this to other higher level units, notably in Unit 8. The reason I'm trying to underscore this is because if you get an essay question, lots of you now are quite confident about discussing transcription and translation, but you might have part of a question which is connecting this with the idea of flowering plants. So the other statements are mostly self-explanatory, I think, but again, I want to jump down to the statements S1 and S2 down here. And the requirement here is that you are expected to make drawings. And remember that if you're asked to draw something, you are always asked to label it. And again, don't just skip over this. Make sure that you can do a proper job of that at a higher level standard. Finally, we've got this last one, S3. Ensure that you are really, really clear with this one about the distinction between germination of a seed and then growth of the plant. And it's a very common misconceptions in the language that's used sometimes when you use them interchangeably and they're not. So germination of the seed is different from growth, and we'll come back to that later on. For this slide, what I would like you to do is just pause the video in a moment and see if you are capable of recalling what you know from IGCSE and using these labels here for this particular diagram. So drawing of half views of animal pollinated flowers. The animal that you're familiar with is insect pollinated flowers. It's not always an insect, but this is what you're expected to draw and you're expected to label. Remember, it's an angiospermophyta, and you need to recognize the connection there with Unit 5, as already said. So pause the video, see what you can label. So hopefully you've had some success there and the IGCSE knowledge is embedded. So I'm going to skip now straight down to slide 5, this one here. And what we can see are all the diagrams, the labels rather, on the diagrams. Just to call your attention to this, this is the male part of the plant. The male part of the plant is called the stamen, easy to remember because it's men. And notice this is a symbolic representation of male parts here. So we've got the gamete being produced is the pollen. Be careful, pollen is not a cell. So again, just check in with the language that you're using. Over here, what we've got is the ovary, and we can see that the ovary is made up of a stigma, a style, and then you've got the ovary at the base here with the ovules, which are the gametes of the female. These are individual, they're not individual cells, that the cell is within those. If we move to this slide, We've got an illustration here which looks at pollination. Pollination is the transfer of pollen, and the transfer of pollen, okay, is being noted here by the biotic factor, the B, the vector is carrying the pollen hopefully to another plant so that you've got cross-pollination. And cross-pollination means that you've got two different parent plants, which is promoting variation. And again, connect everything that you know about reproduction, mitosis, meiosis, etc., etc. A word of note and a caution again is that pollination is the movement of the pollen grain, which is a gamete, as opposed to over here we've got seed dispersal, and the seed is the fertilized ovule that's produced, and the seed has all sorts of adaptations that allows it to be dispersed in a variety of ways. And this one here, you can see is the banana. The seeds are inside the banana, and that is taken in by an animal. The animal walks away, digests that banana, and displaces the seed of the banana with ready-made fertilizer elsewhere. You've also got things that are hooked. Okay, so you've got an example here. If you go hiking up in the hills in Hong Kong, you very often find burdock up there, and burdock is stuck to your pants. You go through the wash and it's still stuck to your pants. But the idea here is that we've got a fruit, the seeds at the bottom. The fruit here, this is a fruit we are familiar with. This is also a fruit, and this fruit is adapted for animal dispersal by a hook-like structure. You've also got other mechanisms like wind dispersal and water dispersal denoted here, but just be careful that when you're talking about pollination, you are making that very, very distinct from seed dispersal. But in both cases, these are using biotic factors. You've got a biotic factor here and a biotic factor here and here. And then you've got also abiotic factors, i.e. the wind and the water. And this next slide, pull them up. So what you can see here are a reminder that usually it's insects which are doing the transfer of pollen, but it can be other animals. And again, showing that this idea that the animal and the plant have got a mutualistic relationship. The animal is going to take nectar, and it's also going to contain some pollen there, which contains protein. So it's getting a nutrition award, and the plant is benefiting because it's moving that pollen around. So we've got mutualism and abiotic factor, and that is using animals as vectors to transfer the pollen. Just check in as well that you've got grasses. Grasses can use wind or water to move the pollen. That's less frequent than the animals, but just remember that some flowering plants have got flowers, but they are reduced and they are green, and they're not the brightly colored insect attention-seeking type. So we've got wind pollinated flowers, and we've got insect pollinated flowers, or more generally animal pollinated flowers. And remember that birds and bats and insects can all have a role. Reminder of mutualism as a term that we need to know. Now, this is where it starts to get a little bit more involved. I would urge you again to pause the video here, pull up BioNinja, and within BioNinja there's a video link which is a scan and electron micrograph which is done as a kind of time-lapse photography piece. It's amazing to see it, but what that's talking about is this statement up here. We've got floral initiation, so the development of the flower is caused in the apical cells. So remember, the apical cells are the meristematic cells, and we're talking about cells here which are in the bud of a flower. And what happens with these cells specifically is that you are switching on genes. And if you're switching on the genes, you've got transcription, you've got translation, and the protein that's produced as a consequence of that can bring about flowering. And we're going to dig into this in a lot more detail in just a moment. This idea of photoperiodism is important. It's the hours of light compared to the hours of darkness that brings about the initiation of changes in the gene expression of these apical cells. So just make sure that you are clear and that you've revised transcription and translation alongside this particular example here. Now, this slide is useful. I would suggest you have a good look at this, but I'm going to jump down to this slide, which I've stolen from BioNinja again. It talks about the idea here that photoperiodism, photolight period, is a timepiece. So how much light is required for flowering is a bit of a misnomer, because it's the hours of darkness that matter the most. Now, what you need to appreciate here, plants need to flower at the appropriate time of the year. So it's no good a plant flowering if the insect that's required to move that pollen is in hibernation. So you've got to have the idea that the plant and the flower of the plant needs to flower at the correct time so that the vector can be successful in terms of moving that pollen. This idea of photoperiodism relies on the fact that plants are sensitive to light. So in the leaf of the plant, what you've got is this, which is what's represented here, something called a phytochrome molecule. And a phytochrome molecule, it's a protein molecule, it's a pigment molecule, and it changes its structure when light falls on it. And you've got two different forms of this phytochrome. So this one here is showing you phytochrome red, and over here you've got phytochrome far red. Now in regular daylight, you've got quite high quantities of this 660 nanometer red light. So in daylight, this form will move to become this form. You can artificially stimulate that process to take place, and it's done very quickly if it's done artificially by flashing red light. So if you flash pure red light, it will switch from this form to this form, but in normal daylight, it will change from this form to this form, but lots slower. So again, looking at this one, it will convert itself back to this form. How does it do that? Well, we can artificially make this quick transformation using far red light, which we're talking here about 730 nanometers. So if you flick this far red light, it'll change very, very rapidly. But in darkness, it will also move back, and the conversion is much, much slower. Now, the other thing to bear in mind is that this phytochrome is only active when it's in this particular structure. But we need to take care about this. So if you've got a long day, like a summer day, then you're going to get a conversion of this to this form, phytochrome far red. Then in the summertime, the night times are short. So you might have some conversion back, but not too much, and then it's going to be converted back to this form. So if you've got a summer plant and you've got a long day plant, you're going to have a lot of this phytochrome red building up, and that phytochrome red that builds up is going to initiate flowering. So long day plants and short day plants are what we need to consider next, because it can be confusing. So I've tried to make it easier in this way, and we'll flick back and forth. So we've got something called a long day plant. An example that is used later on in this slide is the iris. That's the iris there. And this plant will flower in the summer. It's insect pollinated. And so we've just made mention of the fact that in the summertime, we've got long daylight hours, short nighttime hours, which means that the principal form of phytochrome is going to be phytochrome far red. You're going to have a high quantity of phytochrome far red. And phytochrome far red is the active form. And what that is going to do is it's going to cause a protein to be produced. We're not really certain of the pathway here, but the protein, which is a consequence of being activated by this molecule, is going to act as a promoter to initiate transcription and translation of a protein, which then gets moved to the flower bud, the apical stem, and it causes flowering. Now, I've put a question mark here because there's some suggestion, but very, very little research support for a molecule which can be referred to as florigen. So florigen, take care with using that word. You might come across it in your reading, though, is thought to be the plant hormone that initiates flowering. But we need to bear in mind that we've got high phytochrome far red being produced in the summertime in the leaf. So within the leaf, something needs to happen to create a protein which acts as some kind of promoter for florigen, and then the florigen or otherwise needs to be translocated to the apical part of the flower, and the flower then starts to develop. Now, we've also got something called a short day plant, and short day plants are going to flower when the days are short, i.e. they flower in winter time. So in the summer, you are a long day plant like the iris, high phytochrome far red. It's going to cause all these things to take place, and you're going to cause flowering to take place. If you are a short day plant, you're still going to have high quantities of phytochrome far red. Phytochrome far red is still the active form, but this time it's inhibiting flowering. So the active form far red is going to inhibit the flowering if you are a short day plant.